Assalamualaikum and hi to Dr. Hafiz Burhanuddin. My name is Muhammad Saiful Aizam bin Johan Arifin, S59301. I will present the proposal of my study, which is the abundance and density of phytoplankton in different depths during summer 2022 in Perhentian Island. We will have the short introduction of the phytoplankton. Phytoplankton can be grouped into three small groups, which is dinoflagellate, diatom, and cyanobacteria. All of them under the kingdom of Chromista, but the different under the different phyla. Next we go to the research background of my study. What is phytoplankton? Phytoplankton is the most important organism that act as the producer of the primary productivity in a marine food web. So phytoplankton also responsible to almost 15% of our total of primary production. They also are the main significant in biogeochemical cycle and climate regulation. Phytoplankton also can be found almost all in all water, and phytoplankton distribution and species can be found more in the deep water. How climate change affect the phytoplankton? Climate change will affect the phytoplankton directly through the physiology and indirectly by the water column stratification and the availability of resources such as light and nutrients. Changes in climatic conditions such as changes in temperature and nutrient, it can modify the phytoplankton structure and the taxonomy composition. Last but not least, the extension of the growing section and vertical mixing is the most significant climatic effect on the phytoplankton composition. Vertical mixing is the vital condition in phytoplankton which can affect the availability of light and nutrients. Why Kitaharana is being used is because it's the modern one and the most fastest method we can use to sampling from the very water depth. Kitaharana can easily handle, but in the most old study we can see they use Vendon water sampler, which is take more time, which is we need to take 10 liter of water, then we need to use plankton net in different mesh size, and it takes more time. This is the objective of my study. The first one is to identify the phytoplanktons. Second, to determine the most abundant phytoplankton in Perhentian Island. The third one is to study the greatest density of phytoplankton in Perhentian Island. Last one is to compare the abundance and density in the different depth in Perhentian Island. This is the sampling site of my study, which is in the Perhentian Island. Next I will explain about the methodology of my studies. The first part is the sampling technique. Before we start our sampling, we need to collect the physical parameters such as weather, coordinate, and deploy depth. Next, we will use the Kitaharnet with 100 micron mesh size. We lowered it in the water and vertically. After that, we will use the clinometer to calculate the actual depth of the Kitaharnet. The next one we will use the nets we will retrieve back to the boats, but before we retrieve it we need to rinse it 3 times before we collect the sample. We collect the sample in the label sample bottle. After that we repeat the procedure with the surface water. The second part is the preservation technique. After we collect the sample, we use 80% ethanol to preserve the sample. The volume of the preservative will be 4% of the volume of the sample. After that, we will concentrate the sample by the precipitation processes. Next, we will go to a data analysis. For the cell count and identification, we will use the small counting slide, compound light microscope, and the Leakey table, which we can see the center one is the Leakey table. This will have the 11 times 7 row and column, which is have the different cells and filaments. After we fill the Leakey table, we use the normal identification table, which have kingdom to the scientific name and the number of the individual. For the species density, we use the formula below and tabulate it. The last one is to compare the abundance and the density of phytoplankton. Graph will be plotted using the result from the previous analysis. So we can see here is the example from the Zhang in 2019, which is in different season. So for the expected result, I expected there will be more plantain species identified. From the previous study, we can see 260 taxa in the South China Sea, 70 from the Kuantan Pahang, 163 in the Langkawi Island, and 84 in the Carey Island. The next, I expected that the species under phylum Basilerophyta, which is the item, is the most abundant between other two phylum. Uh, so we can see here 63 from the 70 species have been identified was diatom in the Muhmmad No in 2013. Other than that, the Cetostros crucitum and Cetocross leve were the most abundant species in the Langkawi Island in 2005. Other than that, I expected that a species under phylum Basilerophyta, which is diatom, has the greatest density between other phylum, which is Cyanobacteria and Myozoa. Most of the data from the density on the density of phytoplankton show that the items has the highest density than other phytoplankton and can be found almost every station from the study of Sohaimi in 2019. So the last one is the abundance and density of phytoplankton in sea surface is less than the sea bottom. According to Muhammad Noor in 2013, 70 species is identified at the 0.5 meter below the sea surface and 163 species identified at the bottom sea surface by Sohaimi in 2019. So this is my Gantt chart research activity for my FYP. And this is my reference that I use. That's all from me. Thank you.